So today I'm going to discuss Chapter 7 in my molecular immunity book. Chapter 7 deals with additional T cell signals that are required for T cell activation via the T cell receptor, and so that the whole thing starts off and running. And you know, in the 70s and 80s, we got through. Basically, it took us 20 years to really figure out that in order to trigger the T cell antigen receptor, we needed to have a short peptide antigen bound up into the MHC, major histocompatibility complex gene product on the surface of an so-called antigen presenting cell. And that was then called signal 1. But signal 1 wasn't enough to really activate the T cell. You needed other kinds of things that macrophages, for one, as the major antigen presenting cells, supplied. And one of those things were co-stimulatory cytokines, such as interleukin 1 and interleukin 6 and tumor necrosis factor. Those three cytokines that are also the so-called pro-inflammatory cytokines act as co-stimulatory agents in order to activate the T cell. But this chapter is going to then add on to that. So that happened in the 70s and the 80s. And by the end of the 80s, other reports started to appear that the story was not over yet. And so in the 80s and 90s and into the aughts, the field, when I say we, I mean the field, struggled with what was actually going on with all these so-called co-stimulatory signals. And ultimately, we also identified co-inhibitory signals. And so the alphabet soup got a little bit more complicated. If people were not the cognoscenti, the aficionados and steeped into this business, it all became a little bit too arcane and esoteric for the common immunologist if they didn't want to deal with all that stuff. But anyway, so at the end of the 80s, there were a couple of papers that reported that talked about the second signal for T cell activation, meaning they were talking about the T cell antigen receptor or the MHC peptide stimulation of the T cell antigen receptor, and they sort of just forgot about the co-stimulatory cytokines. Those are that back to the lymphodrek kind of days, whatever. And so they talked about the second signal, and the second signal evolved then from the late 70s into the 80s. What happened was all of the things that dealt with T cell activation ultimately were mediated through activating the expression by the T cells of interleukin 2 as the major cytokine, but also other cytokines and so forth. So that was a sort of an immediate readout, but then what interleukin 2 did was activate the cell to undergo what we called cell cycle progression and then into proliferation, DNA replication, and then proliferation. In 1989, there was a group of investigators, Craig Thompson and Carl June and Howard Young, all sort of got together and simultaneously identified a new surface molecule that appeared on T cells, activated T cells, and ultimately they did that through the generation of monoclonal antibodies that reacted to cell surface T cell molecules, and ultimately they started naming all these molecules or designating them with a nomenclature that I think I talked about before that's called the CD nomenclature. We have CD4s and CD8s and CD3s, and those were the major initial ones. But then we've got up to CD a couple of hundred or 400 by now. So this new molecule that was found reacted with this monoclonal antibody was ultimately numbered CD28, and C and D stand for cluster of differentiation. And they used to have conferences where they would all get together and exchange reagents and decide whether or not they had the same or a different antibody that recognized a different or the same molecule on the surface of the T cell. The CD28, when you use the antibody to CD28 and activated the cells with anti-CD3, which triggers the T cell receptor, get a marked increase in proliferation. It's huge. So that was a big deal, and people got all excited about that. And that was the so-called second signal. They didn't know what it was and what it did, and where it's coming from and where it's going. But ultimately, they started getting into the biochemistry of this whole process, and they found that what happened was that there was a marked increase in the production by the cell of interleukin-2 particularly, but also other cytokines. And they showed that there was the mechanism whereby this happened was that when you triggered the CD28 molecule in addition to the CD3 molecule, there was a marked increase in the rate of transcription of the cytokine, say for IL-2. But there, in addition to that, and that was up about 5 to 10-fold over baseline. But in addition to that, the messenger RNA that came off of the gene then, that was transcribed from the gene, it stayed around a lot longer. There was a stabilization of the message so that it just kept on translating more and more protein, meaning IL-2, and therefore there was a much greater concentration of IL-2 built up, which then drove the proliferation of the T cells. And that was all good, particularly from my standpoint. I said, well, yeah, I knew that because IL-2 is important and so forth and so on. Ultimately, they did more molecular genetics, and they found that the area of the promoter region or the IL-2 gene that was responding to the CD28 signal on the cell surface was a response element that was responding to the REL transcription factor family, and that's also known as NF-kappa B, and that's probably even more famous than REL, but REL was the initial designation for this. And then in the mid-90s, that was in 89, and then 93, and then 94, 95, and 96, Ellen Rothenberg from Caltech essentially defined what was going on with the promoter region of the IL-2 gene, and she defined the fact that there were 3 different regions that were binding 3 different transcription factors. One was called AP1, another one was called NFAT, for nuclear factor of activated T cells, and another one that was the REL element. And if you had all 3 of these simultaneously activated and produced, they would bind to the IL-2 promoter, which would then promote IL-2 transcription and then translation and then protein production. And if you got rid of one of those transcription factors, and you could do so with pharmacology, there were drugs that would block different transcription factors. And one of them, for example, that was discovered in the early 80s was called cyclosporin A, and that would block NFAT. And if you threw that into the mix, NFAT could not participate in the transcription of the IL-2 gene, and therefore you block IL-2 gene transcription and translation, and you wouldn't get any IL-2, and you wouldn't get any proliferation. And cyclosporin was then really the first drug in the 80s that was used in kidney transplants. There was a huge increase in the efficiency of blocking kidney transplant rejection. And steroid hormones, another one, they were shown to block REL, and so forth. And so there were a cadre of drugs that were developed that worked on all 3 of these different transcription factors that essentially would suppress the immune response, and therefore were utilized to block transplant rejection, and so organs could be transplanted, and they went from kidneys were the first, but then they got into lungs and pancreases and hearts and all kinds of things, and the surgeons it basically made for transplant surgery as a subspecialty of surgery. If it hadn't been for those drugs, we wouldn't have transplanted organs today. Then the question became, once CD28 was defined and its molecular mechanism of action was determined and so forth, people started to wonder about, well, how does CD28 get triggered? There must be a molecule on the surface of antigen-presenting cells that activates the CD28 molecule on the surface of the activated T cells. And so there were another group of investigators. These people were Lee Nadler and Gordon Freeman at the Dana-Farber Cancer Institute in Boston, identified a ligand, another molecule that was on the surface, that appeared on the surface of antigen-presenting cells that seemed to be binding and activating CD28. And the other thing is that what they found in their experiments, they found that interferon gamma, which is, as you may remember, is an antiviral molecule, but it's made by lymphocytes. So it's gamma, there's alpha, beta, and so forth. This is gamma. And gamma became defined really as a macrophage activating factor. And one of the things that gamma does to act as in the process of activating the macrophages, it causes a marked increase in the expression of a molecule that Nadler and Freeman named B7. Then in 1991-92, another fellow, Peter Lindsley, who was interested in tumor immunology and cancer immunotherapy, he said, well, maybe the problem is that tumors don't have B7, and therefore they can't activate CD28 on the T cell to make a lot more IL-2, so you don't get a really good immune response. And so he took the genes coding for B7 and he put them into a tumor line, and he then put those tumor cells in the mice, and what he was able to show is that he could take a non-immunogenic tumor that was never rejected by the mice, and if you put B7 into it, then the cells were rejected. So this was 1992. And just to give you an idea of how long it takes for all these things to reach fruition, that was one of the beginnings of what we now know today as cancer immunotherapy. So, and that whole phenomenon of B7 activating CD28 they gave a name to it, and they called it co-stimulation. It was co-stimulation of the T cell antigen receptor, essentially. And then another group of molecules came along that ultimately were called co-inhibitors, just to make things much more complicated for everyone. And so the first one is called, that became into being and also discovered and became very, very important for immunology, is called CTLA4. CTLA4 stands for cytotoxic T lymphocyte late antigen number 4. And it was originally discovered and reported by Pierre Goldstein, who was at in Marseille at a big immunology research center in Lumi, I think is the name of it, in Marseille. And so that was 1987 when he reported that. And then it took him a while. They ultimately cloned the gene encoding for that molecule, and they compared it to other genes and other molecules that are out there. And lo and behold, it was very homologous to CD28. So they just assumed then that, okay, well, it's just another co-stimulatory molecule. Wrong. And it took about 5 years to figure that out. But it was more important, actually, than just another co-stimulator, because this was the first co-inhibitor. It wasn't until 1994, so that's 7 years later after the initial discovery, that it was found that if you activated T cells via the T cell receptor by anti-CD3 antibodies and then threw in anti-CD28 antibodies, that you would get a marked expression of this CTLA4 molecule, but it appeared late, like 3 or 4 days later. And it wasn't clear why it took so long or what the significance of that was. But it was, and that's why they called it CTLA, a late antigen. But then it was found and this made perfect sense to me, is that it wasn't the activation of the T cell receptor, and it wasn't the activation of the CD28 surface molecule that caused the expression of CTLA4. Lo and behold, it was IL-2, again, sort of rearing its ugly head, because you needed both CD3 and CD28 for the cells to make IL-2, and you needed time for that to happen. And then the IL-2 had to be produced and bind to its receptor, and then that activated the expression of CTLA4. But it wasn't until 1994, so that another group of investigators, Craig Thompson, Bluestone, and Lindsley and Freeman, Jeff Bluestone, came up with another antibody to CTLA4 that allowed them to do another series of experiments. And what they showed is that if they blocked CTLA4 with their antibody, then you would get a hyper response. You'd get a much bigger proliferative response, much more IL-2, much more, the whole thing was much more augmented. And they interpreted those findings as evidence of the fact that what CTLA4 really did was it acted as a brake on the system. It was a turn down signal. And if you blocked that signal, then you got more of the positive things. So you got positive things going on and you have negative things going on. And it took a while to sort all that business out. In 1995, then a fellow by the name of Jim Allison, who was out at UC Berkeley, did a series of experiments that showed that that was very much the case, that it delivered a negative signal. And then in 1995, a group of investigators, Arlene Sharp from the Dana-Farber, who was actually married to Gordon Freeman, who was involved in all this stuff. It's a very incestuous kind of a deal. And independently, Craig Thompson, in another series of experiments, and he was at the University of Chicago, they both made CTLA4 knockout mice. So if you knock the gene out of a mouse, what do you suppose happens? Knocked out the gene for CTLA4. Well, what you got were mice you got mice so that they made it through the embryogenesis and so forth, which was a good thing. And they were born and they looked normal. But then within a couple or 3 weeks of birth, they started to get really sick, really sick. And so they just filled up with activated lymphocytes. And they had them all over, they invaded all their organs and so forth and so on. And essentially they were all dead by week 3 or 4. And so that was a very dramatic finding. And actually in that particular finding, as we go on in chapter 8, particularly in the next chapter of this book, you'll see the significance of that. And then a young postdoc in Craig Thompson's lab by the name of Maria Luisa Allegra did a series of experiments that I thought were probably the best experiments done in this field, in this area. And I've told her that, and of course that made her feel really nice. Because what she did was she took an interleukin-2 knockout mouse, and she activated the cells and so forth and so on, and did a series of very, very nice experiments. And she showed so what do you figure? Since IL-2 causes CTLA4 expression, what would happen if you knocked out IL-2? Well, you wouldn't get any CTLA4. And that's exactly what she found. But if she took the cells from these mice that were, and added the IL-2 in vitro, so just give it back, they couldn't make their own, but you can give it to them, then they expressed CTLA4 just fine, thank you very much. And so to me, that really nailed the coffin. That really settled the whole deal. And at that point in time, I thought to myself, well, this looks like a very important series of experiments. But almost at the same time, Jim Allison out at UC Berkeley figured that this was also very important, and he was interested in tumor immunology and tumor immunotherapy. And so he thought, well, if maybe since the CTLA4 is a turn-off signal, if I block that with these antibodies, then maybe the immune system can reject tumors. So he took this system where he took a tumor that was expressing B7 so that it would be immunogenic, and he blocked CTLA4 with an antibody. And the the tumor cells were rejected much better than ever before. And he also then took tumor cells from just, off the shelf kind of things that were normally not rejected. And if he blocked CTLA-4 in the mice, then those tumors were rejected. And so that was 1996, and that was the beginning of so-called checkpoint inhibitor therapy that is today as one of the major breakthroughs in the whole tumor immunotherapy business, and led just last year or the year before Jim Allison to win the Nobel Prize for that. So that was all good, but there was still more to the story. And because almost simultaneously, so that all happened in the 90s, really. It took a decade to sort all that out and to realize the significance of those findings, these co-stimulators, co-inhibitors. In 1992, there was a group from Kyoto University in Japan, which is led by a fellow by the name of Tasuka Honjo. And they were looking for genes activated that were expressed when cells were under the process of programmed cell death. And it's a whole new our whole process I think I went into in terms of the mechanism of tumor cell cytolysis and so forth. So they were cloning genes that they thought were at least temporarily associated with programmed cell death. And they came up with a molecule, with a gene that encoded a surface molecule that appeared on activated T cells. And they named this PD1, programmed death 1 gene. It took them a couple of years, so that was 1992. It wasn't until 1996 they were able to, get enough of the gene product and so forth to get an antibody to this thing. And they found that when they activated T cells through CD3, through the T cell receptor, that all of a sudden this molecule appeared on the cell surface, this PD1 molecule. And that was in 1996. It wasn't until 2 years later in 1998, that everybody's doing the same thing. So first thing they said, they wanted to know what happened if they knocked out this gene. So they knocked out the PD1 gene. And lo and behold, the mice got sick in a similar fashion as the CTLA-4 knockout. But they didn't get sick as fast. It took them, they lived for 6 weeks, rather than being dead at 3 or 4 weeks. And they were only mildly sick. They had a doubling in the size of the spleen and the lymph nodes and so forth. And the other strange thing about it is that they had both the B cells as well as the T cells seemed to be proliferating, hyperproliferating essentially. And then they knocked the gene out of another mouse strain that lived even longer. But ultimately, those at 6 months of age should tell you that mice reach puberty at 5 weeks of age, and mice live about 2 years essentially. So these mice at 6 months of age, they were clearly adults. And they developed a whole syndrome that looked like what in humans, an autoimmune disease that humans get, that is called systemic lupus erythematosus. It took me a long time to be able to say that, just roll it off my tongue like that. I used to have one of my, it reminds me of one of my mentors that would when I was in medical school, and he'd always come up with these words that he could say so well, and one of them was Shuchikamuchi fever. I never knew what Shuchikamuchi fever was, but I practiced it so that I could say it just as well as he did. So this wasn't the same kind of, it was a similar kind of a phenomenon that happened when you knocked out PD1, but it wasn't the same as the CTLA-4. And so the investigators that were interested in this, really some of the same people that were into the CTLA-4 business, they wondered whether or not the ligand, the stimulator of PD1, was a B7 family member. And so they looked for other B7 molecules in the gene libraries and so forth, and did searches, and came up with a new ligand, which they called PDL1, PD ligand number 1. They found that this PDL1 would be expressed on macrophages, and particularly if you gave them interferon gamma, they would have a whole bunch of PD-1L1 molecules on them. So that the whole thing seemed to make sense then. And if you then activated cells in the test tube with anti-CD3 and had macrophages there and added either an anti-PD-1 antibody, essentially what would happen was it would tone everything down. So it was giving a negative signal, just like CTLA-4 was giving a negative signal. And that was, they first showed that in 2000, and then 2001 they did more experiments. Gordon Freeman together with Hanjo, and then another group, Carter and Carreño, who really contributed nice things, because they showed that the thing is that the PD-1 molecule was really not expressed. It was undetectable on the macrophages unless they were activated. And they showed that the activation of PD-1 on the surface of the T cell with the PD-L1 would essentially inhibit IL-2 production. So it was a very similar story to CTLA-4, but it was a little bit different because of it was qualitatively different. And so both of these, and so then ultimately what happened was that tumor experiments were done by Hanjo and others, just like Allison had done with the CTLA-4. Lo and behold, if you block PD-1, you also got a marked increase in an anti-tumor immune response, which led to Hanjo sharing the Nobel Prize with Allison just a couple of years ago. But I think that in sort of rounding out these co-stimulatory molecules and co-inhibitor molecules and their ligands, and the whole thing was that obviously this is a very complex process of T cell activation, of activating the T cell immune response, and it's multifaceted, factorial, but and maybe I'm biased about this whole thing, because if you read through all of these papers and all the things that took 2 decades, 20 years essentially, to really unfold and be so that I could tell you about this in a half an hour or so. Interleukin-2 is the conductor of the symphony, because without interleukin-2 you don't get anything. You don't get the co-stimulation, and you don't get the co-inhibition, and you don't get tumor rejection, essentially, because the reason you reject the tumor is that you get a hyperimmune response over normal. And so I'll stop there.